Right, we're continuing on with the digestion topic paper. So here you've got a table where you've got the name of the process and the description. So the first one is food enters the mouth. It's called ingestion. Digestion. So that goes in through the mouth, right? Digestion. Right, just on the side here, because we're learning at this moment in time. What does that do? That it softens food. It breaks it down. What's the benefit of that? To increase the surface area. Right, for what? For the amylase and to act. And what did we say amylase was? Amylase was right, okay, it's an enzyme which breaks down starch into maltose. Remember, ose is a sugar, ACE is an enzyme generally. Okay. So digestion is the breakdown of large insoluble molecules into small soluble molecules. I'll try to write like this rather than joined up. Right, so when small food molecules move from the small intestine into the blood, it's called absorption. Absorption. Small food molecules that are in the blood are used to build larger molecules. That's called assimilation. Assimilation. That's when the food that you've just eaten becomes part of your body, becomes cells. Digestion is the removal of undigested food out of or through the anus. Okay, that's part one. Right. Describe the process of digestion in the mouth. Okay, 3 markers. So it's in the mouth. What happens in the mouth? You get mechanical breakdown or digestion. Mechanical breakdown of food into smaller pieces. Right, what does that do? That increases surface area for enzymes to act. Right, so the saliva or salivary gland produces amylase. Right? Produce amylase, which is an enzyme which breaks down starch. Let me write that properly. It breaks down starch into maltose. So remember, ose is a sugar. These are carbs, carbohydrates. Okay, let's go. Right, you've got two food tests here. One's with iodine. If you add iodine, it turns black blue, and if you add Benedict solution, it turns brick red. Right, concluded that they're both carbs. Okay, so remember we said carbs, carbohydrate. Right, you've got starch, and you've got glucose. You've got lipids, those are fats and oils. Fats are solid at room temperature, oils are liquid at room temperature. And when lipids are broken down, they're broken down into two molecules, glycerol and something called a fatty acid. Right. Protein. Protein breaks down into, and this is the key thing, amino acids. There's other steps in between, but the one that I know or I've seen in the mark scheme a lot is peptides. And then peptidase breaks peptides into amino acids. Right. Iodine. When it turns something black blue, you've got starch. Starch is present. Why? Because why is starch present? Because the sample turns black blue. Do you agree with this? Yes. Let's put that first. The sample turns black blue when iodine is added. Right, the next one is Benedict solution. When Benedict solution, so that's starch. Okay, let's start with control. Right, glucose is also present because, let's just add this for completeness sake, because sample turned brick red. Okay, that's the key word, brick red. Don't just put red. When Benedict solution was added. Benedict's solution was added. Okay, that's that done. Right. In digestion, large food molecules are broken down into small food molecules by enzymes. What's an enzyme? An enzyme is a catalyst. And what does a catalyst do? It speeds up reactions. Right, at this level, we're just going to leave it like that. In chemistry, you'll have to add a bit more. Right, so starch starch is the molecule. You use amylase and it breaks that down into glucose. Remember, starch breaks down into glucose. Protease is to do with proteins. Remember the ases. Right, so it'll break down protein, and what did we say? It breaks it down to, we can use, right, I prefer to write amino acid here. We can check if it's if it allows as in peptides. Yeah, it allows all three. You can put peptides or polypeptides, but amino acid is the safest one. Lipid, remember we said, is look at it as a fat or oil. Right, so you need lipase. Remember the A's? And it'll break that down into which two molecules? Did we do it up here? Yeah, can you see that? Glycerol and fatty acid. Let me just show you that again. Yeah, so that there. Glycerol or fatty acid. There you go. Right. Small food molecules are absorbed in the villi, in the intestines. How are villi adapted to absorb small food molecules? Right. Right, so look, there's your intestine wall. Well, that's the outside. This is the inside, and you got a little projection like this. That's your villi or villus. Now, that's the first thing. Then you've got a little structure inside, lacteal. It's linked to the lymph system, but eventually goes back into the blood. And then you've got we've used the wrong colors. You've got capillaries, a network of capillaries. So, for example, I don't know why I did that. That should have gone up there, and that should have come down here. Right, so you've got a network of capillaries. Capillaries. Okay. That yellow bit inside is the lacteal. And then you've got the villus or villi. Right. So let's do it this way. So what you've got is the villi increase the surface area for more efficient absorption. Right, that's the first bit. You have a lacteal in the center. And what does that do? That absorbs lipids and it enters the lymphatic system. Right, you've got villi, lacteal. You have a network of capillaries, which are one cell thick. So that means you have a short diffusion distance or pathway. So it makes for a higher concentration rate. Also, blood flow maintains the concentration gradient. So you've got a few things there. So you've got surface area, you've got lacteal, which absorbs the lipids, you've got a network of capillaries, which are one cell thick, short diffusion distance, and then you've got blood flow, which maintains the concentration gradient. Basically, all that means is, look, let's do it here. There's your capillary. Here is your intestine. So look, your food's here. So your food's here. That needs to get into here. So what happens, it starts diffusing in. Now, if the blood wasn't flowing that way, what would happen is eventually the concentration outside would be the same as the concentration inside, and diffusion would stop. You would stop absorbing the food. But because the blood's flowing, what happens is that disappears. So the concentration gradient continues. So some more of it moves in, and it just keeps maintaining that. And also another adaptation is that it has to be permeable. So look, that one and that one, let's get rid of this and say the villi are permeable to allow small food molecules through. Small food molecules through. There you go. Is that done? Good. Right, okay. Let's look at this. What happens to fish protein in the human stomach? Ooh, didn't we just do that earlier? Yeah, we did in question 2. So what happens to protein from the pigeon meat? So protein in the hawk's stomach, and it'll be the same thing. So you've got, the stomach contains hydrochloric acid, it secretes pepsin, the pepsin optimum pH is low, and digests the protein into peptides or amino acids. There you go. So that's what we need to write. So you can see it's just repetition. So what happens is the stomach contains hydrochloric acid. So look, I've used the chemical formula for the hydrochloric acid, and you should know this for chemistry anyway. H and then hydrogen and chlorine. Hydrochloric acid contains hydrochloric acid which has a low pH. Right, that means and then the hydrochloric acid kills off pathogens. It's a 4 marker, kills off pathogens. What did you say pathogen was? Pathogen. We said it was a disease causing microorganism. Right, the HCl kills off pathogens. Okay, and then the stomach wall secretes. What does it secrete? It secretes pepsin, which digests protein into peptides. You can also put amino acids, but we'll leave it like that. Right. What we'll do is, this is about 20 minutes long, so we'll finish this off here. and we can start the other questions later. Right, we've got to complete this table. So the protein is hemoglobin, something, insulin, and something else. Right, the function of the molecule. So remember, most enzymes are made, I think all enzymes are made out of protein. And where does it take effect? So hemoglobin, we know, is to do with oxygen and red blood cells. So it transports or binds to, transports oxygen around body. And remember what we said about hemoglobin? It's this shape. Let me just, oh my god, yeah, okay, it's a biconcave shape. Cave shape. What does that do? It increases surface area for oxygen to diffuse in. Oxygen to diffuse. Right, it's got no nucleus. Look, normal cells have a nucleus. This one doesn't. So there's no nucleus. That means there is more room for, if I can get this right, hemoglobin or globin. So there's more capacity to carry oxygen around the body. Right, to digest starch in the salivary gland, remember what we said, it's amylase. Right, insulin, what insulin does is when you've eaten, you've got loads of glucose. So this is glucose in your blood. So that's your blood. And you've got too much and you don't need it at the moment. So what insulin does, it grabs these, joins them together into a long insoluble. So these are soluble. That's glucose. After you've eaten, it joins them together into an insoluble long molecule called glycogen, and it stores it in the liver. You've got to remember that. Right, okay, so insulin, where is it? So the function is to turn glucose into glycogen. Place where that is made, right? It's made in the pancreas. So remember, in the pancreas, you've not only got insulin, you've also got glucagon. Right, something that binds to antigens on pathogens. So antigens, think of a white blood cell. It's a lymphocyte that produces an antibody. It produces an antibody. It's produced in the it's produced by the lymphocyte, but it's produced in the bone marrow. Yeah, so what we'll do is we will not put that there. The actual protein molecule is an antibody and it's produced by the lymphocyte. So we want to write it that way. Right, okay, that's almost 25 minutes long. We will do question 9, 10, 11, oh my god, 12. Oh my god, 12. Right, okay, we can do that in another video. That's the next one. Inshallah. Okay.